Okay, thanks for coming back today. That first day meltdown, everything seems to be working today, so we'll keep our fingers crossed. I feel like maybe there was some things that I meant to say that I probably didn't in terms of the syllabus and just getting started. It looks like a number of you are into Top Hat already. A lot of you have started the first homework assignment in Top Hat. So the one question that a few of you have asked is, hey, I got that question, that fill in the blank question right, and it didn't give me credit for it. The reason is that I have to go in apparently and do that all manually. Or if you put a space in and, you know, or spell it different, or there's all sorts of things that I'm going to have to go and do that manually. So I'm thinking that I may not actually do that until it's the due date, and then I can go through and do everybody's at once, and I don't forget to grade some and not others. But I may be able to do that. There might be a way that I can kind of do that more as we go. But so that's one thing. If you do have, you know, questions about homework, I'd say there are some questions that I'm not totally in love with that are on there. A lot of them I specifically put there though. So I say 90% of them are good questions. There's a few in there that I look back at and I think, oh, why did I put that in there? So bear with me. Again, if there's questions that you think are wrong or otherwise have an issue with, don't be afraid to come talk to me about that. Again, we're shooting for 80% accuracy. There is some participation points kind of built in there to kind of soften that up a little bit. We'll kind of see how it goes and may need to, you know, adjust things as we go. But I'm hopeful that you can all get all those 60 homework points. Again, that will be kind of added in at the end of the semester. So, all right, don't be afraid to stop and ask questions as we go along. I can clarify things, or if, you know, just something occurs to you that you wonder about, we can stop and do that. But last time we started with a little history. Hopefully I conveyed at least the people that I want you to kind of know the basics about what they did. And then we started into a little taxonomy lesson, which to me seems a little bit out of order. Next, we're going to talk about the kinds of organisms that we're going to study in this class. We'll try to make it flow here a little bit. And then we'll get into a little bit on how we grow microbes today as well. We'll see how far we get. But where we left off, we're talking about taxonomy, and I mentioned that this is a science that is continually changing. We're continually learning more about organisms, and we kind of understand how they're related to each other better. So this is something that even today we are constantly regrouping organisms and changing their names, which is really kind of annoying when you've been working with an organism for a long time and suddenly the name has changed. But we're all kind of doing our best. So where we left off last time, we were talking about how we've added some kingdoms along the way to Linnaeus's original, you know, phylum, kingdom phylum scheme. And you don't have to have that memorized. You should kind of understand, hey, at least Linnaeus had the kingdoms be the highest. We're going to talk about how we've actually added a unit of taxa higher than that now. coming down to genus and species being kind of the most specific. But today we are especially interested in not just obviously what organisms look like and their characteristics. We're especially even more interested in the phylogenetic relationships between organisms. And that's a fancy way to say evolutionary relationships. Okay, so we want to try to understand when we are grouping organisms together, we want to understand which ones have a closest common ancestor. Okay, that's you might have seen these big phylogenetic charts. I'm going to show you one in a minute. But so Carl Woese and George Fox was a student in his lab that we give credit to as well. Back in the 1970s, they were interested in the relatedness among all organisms, and they decided to sequence ribosomal RNA in order to determine evolutionary relationships between organisms. So you might be thinking, well, why didn't they just use DNA? DNA is a much more complex molecule than ribosomal RNA. So really sequencing at that time was really a slow, cumbersome process. It took a long time. I just was reading before class that if they would have decided to use DNA to get this data, they probably would have taken them 10 years longer than if they would sequence ribosomal RNA. Where do we find ribosomal RNA? Not a trick question. It makes up the ribosome. Okay, and what do ribosomes do in the cell? They make proteins, right? Good. Hopefully learned that 7th grade biology. So ribosomal RNA, that's the main molecule inside the ribosomes of cells, and that's a molecule that's really highly conserved. Because if something goes wrong with the ribosomal RNA, if there's a mutation, then probably the cell is not going to survive because it can't make proteins correctly. Okay, so it's, again, extremely highly conserved. That just means the mutation rate is really, really low. Okay, so seems like a good thing to use. Later on, we have also been able to use DNA to do the same things, but at that time, they used ribosomal RNA. And what they expected when they sequenced many different organisms, their ribosomal RNA, they expected to find 2 basic groups of organisms. Because prokaryotic ribosomes, remember prokaryotes don't have a membrane-bound nucleus. Prokaryotic ribosomes have a slightly different structure than eukaryotic ribosomes. Okay, that's why we can take some antibiotics that target protein synthesis of bacteria, because their ribosomes are a little bit different than our ribosomes. Okay, so he thought that he would find 2 major groups, but surprisingly, he found that there was 3 main groups of that ribosomal RNA. So that was a really kind of interesting finding. And he suggested that there needs to be a level above kingdom to account for that. Okay, so he suggested calling that taxa domains. So he suggested calling them the bacteria, the archaea, and the eukarya. And of course, the bacteria and the archaea are the prokaryotes, and the eukarya are the eukaryotic organisms. Okay, so Carl Woese, he's kind of thought of the one that created this name, this archaea, or archaebacteria, it's also called. All right, so I think it was in maybe not till 1990 or so, that he published his phylogenetic tree of life. And so this mostly probably looks pretty unfamiliar to you. It does to me too, but if you look at this, you are here, okay? So we belong over here in the eukarya, we're in the animal branch, okay? And again, these are the domains, and with the bacteria and the archaea being prokaryotic cells and eukarya being eukaryotic. All of these other names that you'll see on here, those are correctly called kingdoms now, right? So we went from plants, fungi, protista, Monera to this system. So it's not familiar to a lot of people. We still use those old names because they're more recognizable. But if you're, looking at, again, genetic relatedness, evolutionary relatedness, then this is what we think kind of the best way to represent that. So a lot of these over here on the eukarya are what we used to call protista, protozoa. We've got our fungi and plants here as well. The archaea are mostly thought of as kind of extreme organisms. It's not necessarily true. You probably have some archaea in your body right now. So we often think of them like the halophiles. Those are the salt-loving organisms. You find them like in the Great Salt Lake or the Dead Sea. You've got some weird stuff like some organisms that produce methane. So they're like in the stomach rumens of cows, for example. There's other organisms that have to, they're thermophiles. They have to live at high temperatures in order to live and metabolize, like organisms you would find in the hot springs at Yellowstone, for example. So there's some weird stuff in there. Most of what we're going to talk about in here are in the bacteria domain. So mostly our gram-positives, the proteobacteria are kind of more the other run-of-the-mill bacteria, the gram-negatives. We'll talk about what gram-positive and gram-negative means soon. One other thing that's really surprising about this phylogenetic tree, if you look at the closest common ancestor of the archaea here, they share a closest common ancestor with the eukaryotes, which is not what you would expect. You would think the bacteria and the archaea, because they're both prokaryotes, to kind of be more closely related. So that's kind of a surprising thing. The archaea share some characteristics with the eukarya, especially with kind of the way their genetics, the way they reproduce. A lot of their metabolism, they're more like bacteria. They have some structures that are unique to them. So we will mention them from time to time, more so concerned in this class with bacteria. We spend more time there. Okay. Okay. All right. So, a little bit more about thinking about identification, classification. We use a lot of other things rather than sequencing ribosomal RNA. Okay, so when you're in microlab, and I know not all of you are in microlab right now, but a lot of you are, so I kind of try to make some ties in with lab. I will never be testing you in here over anything that I know that you've only done in lab, so don't worry about that. But when you're in lab, you're going to be tasked with figuring out what kind of bacteria you're dealing with. And there's a lot of ways that we use to do that. We might look at the cells under the microscope. That doesn't give us a lot of information. We can find out other things about bacteria, like do they use a particular nutrient? In other words, do they have the enzymes to be able to utilize that nutrient? Can they break that disaccharide lactose into its monosaccharides to metabolize it? When they metabolize a sugar, do they produce a gas? And in these tubes right here, there's actually a little inverted tube in there. And this organism is, you can see some gas caught in that tube. Okay, so that's just another way to characterize the organism. Does it make a gas when it metabolizes the sugar? Sometimes we might have some kind of an indicator. This is a test for organisms that can make hydrogen sulfide, a gas. Can the organism make a particular enzyme? Less often is it sensitive to some kind of antibiotic. This is a test that you'll do in lab. You can see some of these have a big zone around that antibiotic disc. It's very sensitive to it. It's killed off easily by it. So there's a lot of different ways that we can characterize organism, and that helps us identify them and put them into groups. If you want a authoritative source on the characteristics of any one organism, E. coli, for example, you would go to Bergey's manual. And I think we have a hard copy of this up in lab. It's actually several different volumes. You can access it online, but it's kind of the consensus of thoughts on every organism and how we think they behave and the things that we think they can do and how we classify them. So, all right. So questions about taxonomy, we'll probably be kind of revisiting that now and again. The last part of chapter 1, and I realized when I was going through this, some of these figures, like this one, I have this is from like one source in the OpenStax, and then when we changed our textbook a little bit, the figure numbers didn't quite match up. So I'll try to fix that as we go forward, but you'll find some of those figures in chapter 1. The third part of chapter 1 is just, hey, these are the kinds of organisms that we're going to study this semester. So thinking a little bit of an overview here, more later on most of these organisms. We're mostly concerned with prokaryotes. And like I said, that concerns the two domains, bacteria and archaea. We'll talk a little bit more about the difference between those two eventually. The eukaryotes that we're concerned with, again, when we're studying microorganisms, that means organisms that are too small to be seen with the naked eye. Most eukaryotes are much larger organisms, but the protists, those are microscopic. Some algae are microscopic, some are quite large, actually. Their algae are technically considered protists. Of course, lots of fungi are large. There's some microscopic ones as well. Helminths are mostly parasitic worms. And the life cycle of some of those parasitic worms involves microscopic parts like eggs. So we kind of include them here. And then, of course, we're going to spend some time on viruses. They are considered acellular, meaning that A prefix means not or against. Okay, they don't have a true cell. Okay, they have maybe some of the parts that a cell would have, but not considered a cell structure. All right, so to study these organisms, it's helpful to have a little bit of a sense of what size we're thinking about. We need to probably introduce some units that maybe you haven't used to measure anything ever before, because it's really helpful to have a unit of measure that's actually a little smaller than whatever you're trying to measure, right? If we try to use a meter to measure a bacterial cell, it's going to be really cumbersome. Okay, so obviously a millimeter, this is 0.001 of a meter. Whoops, a meter. Okay, so we can still see a millimeter. So if you think to a ruler, a millimeter is 1/10 of a centimeter. Okay, so we can see that with the naked eye. We start to get a little smaller than that, and that gets pretty difficult. If we move the decimal plate place 3 more places to the left, we're going to get to this unit. Again, this is 0.00001 of a meter. Otherwise, 1 times 10 to the -6th meter. Okay, if you see that negative exponent, that means it's less than 1. Okay, sometimes I see that get mixed up a little bit. So a micron or a micrometer is that Greek mu means micro or millionth. That's a pretty useful unit because a bacterial cell on average is about a micrometer. So that's useful. Eukaryotic cells are going to be anywhere from maybe 10 to 100 times the size of a bacterial cell. It's helpful to have an even smaller unit to measure viruses. 3 more places to the left, or a billionth of a meter is a nanometer. I'll write that up here, nanometer. And that's 1 times 10 to the negative 9th of a meter. So that's more useful to measure kind of parts of cells. A lot of viruses are going to be that small. Some viruses, you can see a smallpox virus is almost as big as what maybe a regular bacterial cell would be, but there's a lot of variation there. Okay, so mostly I kind of wrote over this here. A light microscope, kind of a normal bacterial cell, that's pretty much the limits of what we're going to see with a light microscope. And then we need an electron microscope to see anything much smaller than that. Okay, so we'll talk more about our kinds of microscopy as we move forward. Oh, and the textbook actually has this kind of neat link in it, which I'll try to open here, just to kind of give you a nice idea of, let's see if I can, oops. Okay, so it starts out, here we go. The head of a pin, right? So you can kind of barely make out that human hair. If you go up, magnify a little bit more, then you can see that dust mite. Okay, so those of you that have indoor allergies are probably allergic to the feces of dust mites. They're pretty small. You're not gonna be able to make those out just by looking for them. Okay, and we magnify some more. That large yellow thing is a grain of pollen. The purple there, I believe, is a lymphocyte. All right, so that's one of a eukaryotic cell. The red are the red blood cells. We get a little smaller there. Whoop, that pink is a yeast. Okay, and then we get a little smaller. Those green rods are E. coli. The yellow are Staphylococcus. That purple there is an Ebola virus, and even smaller than that are some little rhinoviruses. So, kind of interesting. It's interesting people to kind of help you. There is a huge difference in just the size, even just of microbes. Okay. All right, so again, just a little overview. Of course, prokaryotic cells, like I mentioned, they lack a membrane-bound nucleus. They still have DNA, obviously, it's just a little bit more loosely organized. There's not a membrane around it. Prokaryotic cells like bacteria don't have organelles like lysosomes or endoplasmic reticulum. And there are differences between that bacteria and archaea domain that I alluded to, nothing really specific yet. One thing that I didn't mention, as far as we know, archaea do not cause disease. Okay, so that's really the main reason that we're not as interested in them for this class. Okay, we maybe just have yet to discover some. Like I said, we think there are archaea that live normally in our body, but as far as we know, there aren't any that cause disease. Okay, so I just included some pictures. You can't tell by looking at a prokaryotic cell if it's an archaea or a bacteria. You have to know more about it than that. This picture on the left there is kind of an oddly shaped cell. We're going to look at some common shapes of prokaryotic cells here pretty soon. But that's a bacteria, actually. Here's a staphylococcus. You'll be seeing a lot of those if you're in lab. And here's an example of an archaea, a methanogen. Again, these are organisms that actually produce methane. So here's some commonly shaped prokaryotic cells. So just a little terminology that you'll be getting familiar with. A coccus is a round cell. Okay, so, and that's built into a lot of bacterial names, staphylococcus. Okay, you know that's a round cell because that's built right into the name there. You're going to be seeing a lot of cocci and a lot of bacilli if you're in lab. Bacilli meaning rod-shaped organisms. Okay. So you get familiar with that. And then a coccobacillus, it's not perfectly round. It's really kind of too short to be considered a true rod. Somewhere between a cocci, a round cell, and a rod. That's the coccobacillus. Vibrio, these are kind of comma-shaped organisms. Cholera is an example of an infection caused by a vibrio. And then we have our helical-shaped organisms, either the spirillum or the spirochetes. An example of a spirillum is Helicobacter pylori. It's that organism that is responsible for a lot of stomach ulcers. Spirochetes, things like the organism that causes syphilis or the organism that causes Lyme disease are spirochetes. We're going to pick apart the difference. Both of those spirillums and spirochetes actually move in a corkscrew-like manner. The reason that they move that way, they both have a very different kind of flagella that propels them. So we're going to look a little bit later at the differences between those two. And there are actually some prokaryotic cells that don't really have any distinct shape because of maybe they don't have a cell wall, for instance, and or they may just look a little bit different depending on the environment that they're in. So there's always exceptions with everything, right? Some examples of eukaryotes that we'll be looking a little bit more closely at. This picture here is Penicillium chrysogenum. So that's the mold that we harvest the antibiotic penicillin from. Over here is some baker's yeast. They ferment, give us alcohol. We'll look a little bit more closely. There are some disease-causing fungi. We don't really talk about that too much in this course, but we do have one lecture that we look a little bit more closely at fungal structures. The protists, you've heard of protists like amoeba or paramecium. They are ubiquitous, especially in water, bodies of water. There are some disease-causing protists, one in particular that we'll look more closely at, malaria. And then algae are considered protists. They don't cause disease unless you get an overgrowth of a certain kind of algae. You might have heard about that happening, from time to time in Florida. But marine algae are called diatoms, have very kind of unique cell walls. Again, so we don't talk a lot about algae. They're very important, but we just don't spend a lot of time on them in this class. And I just mentioned the helminth worms. Obviously, most of them are not microscopic. The tapeworm can get up to 10 meters long, just like so disgusting. We do have a parasitic class that's taught every fall by Dr. Graham. Great class, sounds really interesting, but again, not really the focus of this class here. So if you're interested in helminth worms, take parasitology. Viruses. We all know a lot more about viruses than what we did a couple years ago. Like I said, they do not have a true cell structure. They infect all types of cells. So I should mention they have to be in a living cell in order to reproduce. You probably knew that already. They do infect all types of cells. Obviously, viruses infect us, they infect animals, they infect plants. Less so fungi, although I think there's a little bit of that. There are viruses that infect bacteria. This is an example of a bacterial cell being infected by some viruses. There are even viruses that infect other viruses. Mind-blowing. That the viruses that are infected have to be in a living cell for that to happen, but that's fairly recent discovery. And this is a SARS coronavirus here. I've had that picture in my notes for a long time. Actually, just an example of a virus. Viruses either have their nucleic acid is either DNA or RNA. They never have both. Okay, so that was another mind-blowing thing when we found out here's this thing that can reproduce that doesn't have DNA. It only has RNA. Okay. So again, much more about viruses later. Here's a question though, where do viruses fall in that, let me just back up just a second, in the tree of life here? Where would you put viruses on here? What do you think? I tried to trick you. Yes, they don't go on there. They don't have ribosomes. They don't have any ribosomal RNA sequence. It is still sort of debatable whether you consider whether they're alive again. Most scientists would consider them not because they don't metabolize. But we'll talk about that more later. Right. So we don't have them on here because we won't consider them alive. Good. Let's see. I think that does it for chapter 1. Questions? I would encourage you to go ahead and, if you haven't already, try that chapter 1 homework. Okay, I would try to do it just by what I talked about in class. It's very easy to kind of scroll back up into the textbook there, and you should be able to find the answers without too much trouble. But I would say get on that before life gets busy. All right, I'm going to skip around a little bit here. I'm going to jump ahead to chapter 7, just part of it. We'll come back to the rest of it later. But this matches up well with what you are going to be doing in lab today, or you did this morning in lab today, as well as tomorrow. So we're learning a little bit about growth media. It's really important to be able to grow bacteria in the lab because that really helps us understand them better and test them and figure out their properties. So we're going to kind of just get familiar with some terminology that we use when we're growing bacteria, and a lot of terminology to do with different medias that we use, or collection of nutrients that we use to grow them up. All right, so there's really 2 basic kinds of medias that we use to grow bacteria: liquid medias and solid medias. Okay, so we've had liquid medias for a really long time. And what I mean by media, this is just a collection of nutrients. So nutrients, vitamins, minerals, to support growth of the microbe. When we have a liquid media, it's best if you want to grow up a large quantity of organisms, then a liquid media is your best bet. Okay, so good for growing large quantities of cells. And I should mention, we generally refer to liquid media as broths. Okay, so how do we make up a liquid media? Well, today we usually will buy the powdered form, weigh out the correct amount, put it in a flask, usually bring it up to, maybe a liter or with distilled water, depending on how much you need. We sterilize it and then pour it into whatever we want to use, usually test tubes. So you'll be in lab inoculating a lot of test tubes. Oftentimes the media is kind of a yellowish color, not always, but mostly the broths are. And it will look kind of clear when there's no cells growing in there, when it's sterile. But if we inoculate it with some cells and they start growing up, you can see here, this is looking a little bit cloudy, a lot more cloudy here. Sometimes those cells will kind of settle out to the bottom a little bit, and you might need to kind of finger tap the tube and swirl it around to kind of bring those up to see them. Okay, so any cloudiness in the tube, that's what the cells look like. Okay, so when we grow cells in liquid media, they've got lots of room to move around, lots of access to nutrients. The drawback to using liquid media is that you can't tell if you have only one kind of organism in there or if you have many. Okay, so in other words, you can't tell if you have a pure culture. Okay, and that just means one organism in that test tube. Okay, can't tell by looking at it. All right, so there could be some contamination in there and you wouldn't know it, and that could totally goof up your experiment. Okay, so for many years, when scientists were trying to understand what bacteria were, they just had broth cultures. And it posed a real problem to try to work with just one organism at a time. Somebody in Robert Koch's lab suggested using something called agar to solidify media, and we've been using that ever since. So we actually put agar. First of all, what is agar? This is a polysaccharide that's found in the cell walls of a certain kind of algae, red algae, if you're interested. Okay, and what's special about this polysaccharide is that it's inert, meaning the vast majority of organisms can't metabolize it, can't break it down. And that's what we want, because if organisms broke it down, then that would liquefy the medium again. So we want something that will solidify the medium and that bacteria don't chew up. Yeah. Are there any exists that do chew up agar? There probably are a few exceptions to that. That's a good question. I don't know specifically what they are. I'm just sort of going on this assumption that, yeah, there's an exception to just about everything. So that's a good question. I should search that and find that out. Okay, so basically we will, if we're gonna make some solid media, we usually will just buy that in powdered form, but we could take some nutrient agar medium, add some broth, and then when it's still hot from being sterilized, we could pour it into a Petri dish. So we call those, we call it a plate. Or we could pour it into a test tube and we put them in an angle while it cools off and it hardens. When agar gets down to about 45 degrees Celsius, it will solidify. So we got then a nice, we call these slants. We got a little less surface area to grow our organisms on. Okay, so let's see. So this is a tremendously helpful tool to grow organisms on solid media, because it helps us tell or helps us understand if the culture is pure. Okay, we can tell by looking at the growth. If it all looks the same, then there's probably only one thing on there. Okay, where do we get agar? This is actually something that we just have to collect. So after a big storm, places like Morocco, Japan, Spain, people go out and pick up this certain kind of algae, red algae, and get paid for it. So that's where agar comes from. We've tried to farm it and grow it up that way, and we haven't been successful. So there was worries about 6-7 years ago that we were over collecting it and that we were going to run out of agar, but that hasn't seemed to have been the case. So still have plenty of agar. Okay. All right, so terminology that you'll get familiar with when we say we want to, those two terms, inoculate and incubate, they sound very similar, and sometimes students mix those up. But if we're going to inoculate an organism, that means we're going to put some cells in some media. Okay, so when we do that, we often use something called a loop. Yours will be wire. It won't be plastic like this one. Maybe if you work in industry, they might use disposable loops. But we use them over and over again. You dip your loop into maybe a broth culture, and here they're spreading it on a plate. So it kind of looks like you're not doing anything because you can't see anything on that loop. But remember, what you're transferring, they're microscopic. And when those cells divide over and over and over again, they will usually grow into discrete packets called colonies. Okay, so each colony represents millions of cells. When you get millions and millions of cells, then we can see them with our naked eye. Okay, how do we get them to grow up? We incubate them. We put this plate, for example, usually at a nice warm place that represents their ideal growth temperature. Okay, most of our organisms grow best in the lab at about 35 degrees Celsius. So that's pretty close to body temperature. And that's going to be variable depending on what organism that you're trying to grow. But again, maybe 35 to 37 degrees Celsius for a lot of the run-of-the-mill organisms. Anything that's going to cause disease is going to grow best at body temperature. Okay. Let's see. So if we want to try, if we have the goal of getting or maybe keeping a pure culture, and remember, that means one species growing in the entire test tube or the entire plate, for example. We have 3 ways that we commonly will do that. And the one that you're going to do a ton of in the lab, you're going to make lots of street plates. Okay, so right down here, this is an example of a street plate. If you were in lab this morning already, you know there's a specific way that we want you to streak your bacteria, all with the goal of getting isolated colonies. Okay, so all these little white dots here, those are isolated colonies right here. Okay, it's just a colony that's not touching another colony. Okay, and we want to get lots of isolated colonies, or we want to streak our organism out in such a way as to get lots of isolated colonies, because we know that all the organisms in that one colony are essentially clones of each other. Okay, so each colony, it's all the same kind of cell. And if we look here and, oh, all of these colonies, they look the same, we can be reasonably sure that the whole culture is pure. Okay, if you just had everything growing kind of like what it is down here, you could have several different organisms growing together there, and it wouldn't be obvious just by looking at it. Okay, so that's something, you know, if you're in lab, you'll have more practice doing that. You could also do something called a spread plate, which you'll do later in lab. It's where you drop some bacterial suspension on a plate and you just smear it around with a glass rod. Or a pour plate. I believe we are doing one of those in lab this week, where you add the bacterial suspension into some agar that's still hot enough to be liquid. And then you pour it into a plate, kind of disperse the cells. So don't worry about how to do those for me. I'm just pointing out these are ways that we try to maintain our pure cultures by getting isolated colonies. And of course, when we're manipulating all these cells, we're going to pay attention to aseptic technique. And these are just a set of precautions that you're going to take to try to minimize the chances that something is growing on that plate, for example, that you didn't want growing there. So you're doing things like washing your hands and disinfecting the bench, and you're going to flame your loop, things that I'll leave your lab professor to remind you of, but that's the kind of things that we mean by aseptic technique. Okay, so sometimes students mix up those terms, isolated colonies and pure culture. You can see here we have lots of different colonies on this plate. They all look very different. Okay, so this is not a pure culture, this is a mixed culture. And you can see that you can have a colony that's isolated by itself, but it's definitely still a mixed culture. Okay, so it would have to have all the same kinds of colonies in the same plate. Okay, so just to point out, you know, a lot of the colonies that you're going to see in lab are going to not look too different from each other. They're going to be a lot of white colored bacteria, but there are lots of different colors, lots of different shapes and sizes and edges. I didn't post this, but there are lots of ways, lots of terms that we use to describe those colonies. And, you know, again, I don't want you to memorize it, so I didn't post it, but that helps us, you know, to some extent kind of categorize, or once you start working with a certain organism, you learn to recognize what the colony looks like. So going a little bit beyond probably what a lot of you will talk about in lab, as we go forward in lab, you are going to work with a ton of different medias. Okay, you'll hear terminology describing the different medias that we use. So I want you to be somewhat familiar with that terminology. We can describe media as just being sort of a general all-purpose media, enriched, defined, complex, selective, differential. There's even something called enrichment media that's different than enriched media. So I want to talk about some of that terminology. I'm going to give you examples of those. So don't worry about memorizing what the example is. I want you to try to kind of understand, you know, what it means, for instance, to be a selective media. Okay. So if we say describe something as an all-purpose media, that just means it's kind of a media that supports a wide variety of microbes. There is no media that all bacteria will grow on, unfortunately, because they're so diverse. And we think that we actually only are able to culture a very small number of the actual bacteria that we know that are out there. Okay, so even if you take some all-purpose media, you swab a surface and streak it on the media, only a small number of the bacteria that are actually on this surface are actually going to grow on whatever media that you choose, even if it's an all-purpose one. Okay, but this just means ones that we sort of use all the time in the lab. So one example of that is called nutrient agar. There's some other ones that we'll use, like tryptic soy agar or luria broth. And I wanted to look at the recipe of nutrient agar because it kind of points out, hey, these are some of the things that you see in a lot of different microbiological medias. Peptone, that refers to some digested proteins. Okay, so source of amino acids, source of nitrogen to support bacterial growth. This beef extract or yeast extract, that is some, it just means it's enzymatically digested. so that we have a slew of nutrients in there, carbohydrates, vitamins, minerals. Agar, remember we talked about agar, that's our solidifying agent. Sorry, I didn't write that very legibly. Solidifying agent. Okay, if we add agar, you see agar in there, you know it's a solid media. Salt is added not so it tastes good, but for osmotic balance. We're going to review osmosis very soon, but we want a fairly isotonic medium, right? We don't want water entering bacterial cells too much or too much leaving. Okay, we usually pH it about to neutral. Okay, and that's kind of an all-purpose media that you'll be seeing a lot of. Okay, we are out of time, so we will pick up there on Friday. I'll get into microscopy. Have a good day.